Hi, my name is Chad Sherwood. I worked in the Weaver lab this semester for my applied project, and my project title is the characterization and tracking of immune cell dynamics as a model study for a placenta-on-a-chip platform. The main problem we wanted to address was advancing and understanding early gestation knowledge, and we wanted to do that by seeing our cell viability of the placental cells within a 3D hydrogel, and we wanted to characterize the innate immune cell interactions here, and also look at the pro-inflammatory cytokine secretions. First thing we did was create a 3D hydrogel, and we tested how well these placental trophoblast cells, which are indicated by the jar cells, were able to survive and through an Almar blue assay. And so we saw that the cell viability was great, as an increase of fluorescence was indicative of that, and as well as the metabolic activity. We used this hex cell fluorescence as a control line as well, just to make sure. And this refined our knowledge of understanding that these hydrogels were good enough for these jar placental trophoblasts. This was further confirmation of that, as we had about 80% of the jar cells live after 3 days of incubation, as defined by the live dead confocal microscopy pictures as well. We then tracked how these innate immune cells, which are THP1 cells, they're a monocyte cell, how their migration patterns are affected by suspending these THP1s in media around a jar encapsulated hydrogel. We tracked these through 5 days, and we compared them to a control blank cell as well. As you can see, the dynamics between both of the hydrogels were relatively similar across the 5 days, except on day 5, the jar-containing hydrogels displayed much further migration for the THP1s relative to the blank cells, and also in the average number of cells in the sectional hydrogel pictures, the THP1 cells in the blank hydrogels had a lot more cells inside of the hydrogel by day 5, whereas the THP1 cells in the jar hydrogels displayed a pretty similar pattern of cell counts between the 5 days. The average TNF alpha concentration comparison also displayed some differences between a M0 cell, which is a differentiated THP1 cell, which is into a macrophage cell, and the cytokine secretion for TNF alpha between these showed a slight increase, which is a pro-inflammatory marker, and it showed a slight increase between day 1 and day 3 of incubation between these jar hydrogels, but a significant drop off between the blank hydrogels in day 1 and day 3 of incubation. Further studies here, we wanted to look at more pro-inflammatory cytokines and anti-inflammatory cytokines and how those interact with the jar trophoblasts, and also look at adaptive immune cells as well.